Welcome to G Fun Facts Online. What if the most powerful new weapon in the fight against cancer wasn't a synthetic drug or a high-energy radiation beam, but a living organism? And not just any organism, but one we're all intimately familiar with, and usually for all the wrong reasons. Escherichia coli, better known as E. coli. It sounds like science fiction, but we are on the cusp of a medical revolution where we turn bacteria, our oldest adversaries, into our most sophisticated allies. Today, we're diving deep into the world of synthetic biology to see how scientists are programming microbes to hunt down and destroy cancer. The core problem with many traditional cancer treatments like chemotherapy is that they are blunt instruments. They are designed to kill rapidly dividing cells, which certainly includes cancer cells. But it also includes healthy cells in your hair follicles, your stomach lining, and your bone marrow, leading to the debilitating side effects that are all too common. The holy grail of cancer therapy has always been specificity. How can we attack only the tumor and leave healthy tissue completely unharmed? The answer, it turns out, might be hiding in plain sight, inside the tumor itself. Tumors are not just random clumps of bad cells. They create their own unique, hostile environments. As they grow rapidly, they outstrip their blood supply, creating zones that are low in oxygen or hypoxic. Their chaotic structure also creates pockets that are shielded from the body's immune system. While this environment is tough for our own cells, it turns out to be a five-star resort for certain types of bacteria. For over a century, scientists have observed that some cancer patients who contracted bacterial infections sometimes experienced a surprising, spontaneous remission of their tumors. This was the first clue. But intentionally infecting a sick person with a dangerous pathogen is obviously not a viable treatment. The game changed with the advent of genetic engineering. Scientists realized they didn't have to use dangerous, wild bacteria. They could take a well-understood, non-pathogenic strain of bacteria, like E. coli, and turn it into a programmable, cancer-fighting machine. So how does it work? There are several incredibly clever strategies. The first is turning the bacteria into tiny, living drug factories. Think of it as a Trojan horse. Scientists can engineer E. coli to carry the genetic instructions for a powerful anti-cancer drug. These bacteria are then injected into the bloodstream. Because they are naturally drawn to the unique environment of tumors, they accumulate there in concentrations up to 1,000 times higher than in any other organ. Once they've set up camp inside the tumor, a trigger is activated. This trigger could be the low oxygen environment itself, or an external signal we'll discuss later. Upon activation, the bacteria switch on their internal factories and start pumping out the cancer-killing drug right where it's needed most. This delivers a highly concentrated dose of medicine directly to the target, while sparing the rest of the body from its toxic effects. The results in animal studies are stunning, showing significant tumor shrinkage with minimal side effects. But what if we could get our own bodies to do the heavy lifting? This leads to the second and perhaps even more powerful strategy: using bacteria as a beacon to guide our immune system. One of the reasons cancer is so deadly is its ability to hide from our immune system. It cloaks itself, convincing our body's defenders, like T cells, that it's not a threat. Engineered bacteria can blow this cover wide open. Scientists are programming bacteria to release specific proteins called antigens or chemoattractants once they are inside the tumor. These proteins act like a giant, flashing neon sign that says, "The enemy is right here." This signal does two things. First, it breaks the tumor's immunosuppressive shield. Second, it attracts a flood of immune cells directly to the site. This approach is particularly exciting because it has the potential to supercharge one of today's most promising treatments, CAR T-cell therapy. CAR T therapy involves taking a patient's own T cells, engineering them in a lab to recognize their specific cancer, and reinfusing them. It has worked miracles for blood cancers, but has struggled against solid tumors because the T cells have a hard time physically reaching them and surviving in their hostile environment. But engineered bacteria can solve this. By colonizing the tumor first and sounding the alarm, they essentially prepare the battlefield, creating a welcoming environment for the incoming CAR T cells to do their job. In one study, engineered E. coli were used to release a substance that attracted CAR T cells to solid tumors in mice. The results were dramatic. The combination therapy eliminated tumors, whereas CAR T cells alone had little effect. The bacteria essentially acted as a guide, leading the elite T-cell soldiers straight to their target. The third major strategy is even more direct: engineering bacteria to be kamikaze agents that destroy tumors from within. While E. coli is a favorite lab subject, other bacteria are also being recruited. A notable example is Clostridium novyi, a bacterium that is anaerobic, meaning it can only survive in environments without oxygen. This makes it a perfect candidate for targeting the hypoxic cores of solid tumors. Scientists have created a non-toxic strain called Clostridium novyi NT. When spores of this bacterium are injected into a patient, they circulate harmlessly until they find a low-oxygen tumor. There, they germinate and begin to grow, consuming the cancerous tissue from the inside out. This process creates a massive, localized inflammation, which in turn triggers a powerful system-wide immune response against the cancer. In early clinical trials on both dogs and humans with solid tumors, direct injection of C. novyi NT has led to the complete destruction of the injected tumors and, in some cases, even the regression of other, untreated tumors elsewhere in the body, thanks to the awakened immune system. Now, you might be thinking, this is amazing, but it also sounds a little scary. Are we just letting bacteria run wild in the body? This is where the next level of sophistication comes in: external control. Researchers are not just creating bacteria that act on their own, they're creating bacteria that respond to our commands. One of the most futuristic approaches comes from researchers at Caltech. They've engineered bacteria that can be controlled by sound. They took E. coli and equipped them with genes that produce an anti-cancer drug. But these genes are linked to another set of genes, borrowed from a different species of bacteria, that are sensitive to temperature. The key is that focused ultrasound, the same safe technology used for medical imaging, can be used to heat a very precise location in the body by just a few degrees. So the patient is injected with these sound-controlled bacteria. The bacteria spread throughout the body and colonize the tumors, but they remain dormant. Then, the doctor uses an ultrasound machine to focus sound waves directly on the tumor. This raises the local temperature just enough to activate the thermal switch in the bacteria, which then start producing their drug payload. Turn the ultrasound off, and the drug production stops. This gives doctors an incredible, non-invasive remote control to turn the therapy on and off, and to target it with millimeter precision. A similar approach uses light. Scientists have engineered bacteria that respond to near-infrared light, which can penetrate a few centimeters into biological tissue. The bacteria are designed to produce a light-sensitive molecule. When near-infrared light is shown on the tumor, this molecule generates reactive oxygen species, highly volatile particles that kill nearby cells, including cancer cells. This method, known as photodynamic therapy, becomes incredibly precise when the light-sensitive agents are being produced by bacteria that have already homed in on the tumor. And for getting the bacteria to the right place to begin with, some research teams are even exploring magnetic guidance. By loading bacteria with harmless magnetic iron oxide nanoparticles, they can use external magnetic fields to steer these,